Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Food Safety: How Representative Sampling Can Reduce Your Risk. My name is Nima Rajan, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to those questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for future download. At this point, I'd like to thank Sentry Equipment, who helped to develop the content for this presentation. With proven sampling expertise since 1924, Sentry Products and Services provide business operations the critical insights to optimize process control and product quality. They deliver true representative sampling and analysis techniques to customers around the globe, empowering them to accurately monitor and measure processes for improved production efficiency, output, and safety. Standing behind their commitments, they are determined to tackle any application, anywhere. Now I'd like to introduce our speaker for today's event, John Pobolish. During his 17 years with Sentry Equipment, John has developed significant expertise in specifying, selling, and deploying sampling solutions for food and beverage plants throughout the world. Currently, John is the industry manager for the food and beverage markets, working with clients and industry specialists to develop best practice sampling and monitoring techniques and equipment to optimize processes and food safety compliance. John has co-authored sampling guidance documents and presented numerous papers on sampling equipment for food products from dairy liquids to dry bulk ingredients. John holds a bachelor's degree in mechanical engineering from the Milwaukee School of Engineering and continuing education certificates from the University of Wisconsin-Madison and other programs. He resides in Sussex, Wisconsin with his wife and son. He enjoys outdoor pursuits including hiking, biking, and skiing, and is a member of the National Ski Patrol. John also enjoys exercising the privileges of his commercial pilot certificate. And now, without further ado, I'd like to hand over control to our speaker, John. You may begin when ready.
Good morning and good afternoon, and thank you for that introduction, Nima. Thank all of you for joining our webinar today as well. The topic of our session today is representative sampling and how good sampling methods can aid in reducing risk in food processing. For the sake of this discussion today, I'd like to, or I'm going to assume that most of you are very familiar with current focus on food and safety, primarily Food Safety Modernization Act and associated FDA and USDA guidance in North America. And for those of you in Europe, I'm sure there are corresponding standards that apply. We're going to try to connect how product sampling impacts risk assessment and FSMA compliance. And for the agenda, we'll start out with just some links to food safety, the standard and guidance from government, how we look at mitigating risk in our processes, our food processes, defining representative sampling, product sampling methodology, and then we've got some specific sampling applications to review and some equipment that is used in those. As I developed this presentation, I wanted to formulate a key takeaway. So if you take anything away from this today, my suggestion is that we look at the concept that representative product sampling increases confidence in laboratory data and thereby improves your ability to assess and mitigate risks in your process. So we're all interested, I think, in making sure that what we do is analyzed properly and we mitigate risks that are associated with that. So to get started, I wanted to take some guidance from some of our customer base, and one of the more interesting ones I came across was something published by Nestle Corporation. And basically, in that document, it talks about the safety of food is neither guaranteed nor controlled by sampling or microbiological testing, which we focus on, we know a lot about. But it really comes into the process and a preventative approach. The government's got a lot of, in the industry in general, has a lot of guidance in regards to good hygiene practice, of course, hazard analysis and risk-based preventative controls. Good agricultural practices for the products that come in that we use and process, and then good manufacturing practices. So in section 103 of the federal document the FDA document in food guidance, FSMA, the key takeaways from this first paragraph really is that what our job is to implement preventative controls that minimize or prevent the occurrence of hazards in our process, in our food process. And one of the ways to do that is to monitor the performance of those controls and maintain records of it too. So I'm trying to make a link here between the guidance and the compliance standards and what we do in terms of sampling. If we take a look at the food production chain, the thing that we're talking primarily about today is going to be the inputs to our process. So obviously there are standards that apply to agriculture, distribution of product, and then once the product gets to the retail or to the consumer. But today we're going to be focusing on the inputs to processing and then what we do to those things and the risks that can be associated with that. So probably known to most is going to be the typical pathogens that the laboratories in your facilities are looking for. And the three top ones I wanted to kind of focus on would be Salmonella, Listeria, and E. coli, because that touches quite a few of the product streams that we're familiar with, and certainly our company has customers in all those areas. Of course, we're interested in looking at the finished product and the raw material. So there's two kind of risk categories, I guess, that we want to talk about a little bit, which is the finished product and then the material stream that comes in. And FSMA talks about that, especially as it relates to foreign raw materials that are coming in. So if there's a source of nuts, for example, that come into a process from abroad, there's guidance as to how we make sure that those items don't have a high level of contamination, for example, and so on, and how you document that. So it's very important. So again, many of you may be familiar in detail with bacteriological testing and how you do that specifically, but the key point I wanted to get across here is that you have to start with a very good quality or representative sample of the lot. And if you look below at the asterisk, we're defining a lot as a specified quantity of a product that is manufactured, filled, and packed under the same conditions. So for the purposes of our discussion today, we want to talk about essentially a part of your process that's going to go into one, say, shipment of a product. And one of the go-to examples that I have is peanut butter. It's easy to understand. We all, many of us eat peanut butter, and there's a lot of risks associated with that today, not only in producing it, but in terms of allergies and so on. So if we take nuts, process them into peanut butter and so on, we want to make sure that that one batch that goes and gets palleted, maybe it's several trucks full, that would be what we were considering a lot. It's interesting to note because you want to be able to understand what happened to that batch of product and then be able to track it in the event that there is something that you need to take care of. So going into the additional comments about bacteriological testing, I'm going to refer to bacteriological and analytical methods that the government produces, and it basically states that if you don't have a good sample, the results that you get from your lab are meaningless. They're not trustworthy, and that's what the link really is between taking a good sample and then being confident in the results that you have, and then using that data that you trust to make decisions on the risks and how to control them.
All right, so now it's time. Oh, sorry, John.
Go ahead.
All right, everyone, so now it's time for a poll question. So I have launched your poll question on your screens. The question is, what type of sampling are you doing now? We have 4 options here. Our first option is manual point, B, packaged product, C, automatic, and D, none. I encourage all of the audience to vote on real time, and we'll take a look at the results. So again, the question here is, what type of sampling are you doing now? Manual point, packaged product, automatic, or none? It looks like most of the audience has voted, so let's take a look at the results. So it looks like 43% of the audience has selected manual point, 26% of the audience has selected packaged product, 13% of the audience has selected automatic, and 17% has selected none. What interesting results. Now, we actually have another poll question here. I'm going to be launching that on your screen right now. Now, I encourage the audience to continue voting on real time. The question here is, what sample challenges do you have? Now, we have 5 options for this question. Samples are not representative, too little collected, operator safety, worried about cross-contamination, and other. So one more time, the question here is, what sample challenges do you have? Samples are not representative, too little collected, operator safety, worried about cross-contamination, or others. All right, so now we're going to take a look at the results. It seems that 44% of the audience has selected A, samples are not representative. 4% of the audience has selected B, too little collected. 8% of the audience has selected C, operator safety. 28% of the audience has selected D, worried about cross-contamination. And 16% of the audience has selected E, other. So giving back the control to you, John.
Thank you, Nima. Well, excellent feedback, and I'll try to address these statistics as we go forward. Obviously, we've selected those questions to kind of guide the discussion a little bit, but the replies are quite interesting, and I'll try to address them as we go forward. So I think as we go into the practical parts of physical sampling of the product stream, kind of some basics come into play. And so the idea here about representative sampling is that if you have a lot of material, you want to make sure that you accurately take a smaller portion of that lot so that it can be used in analytical purposes. So essentially you see on the right of the screen 2 blue boxes, and the first one or bigger one with the yellow and white dots on it represent a lot of product, and it's basically 10 items total, or 20% of those are white colored items. So as we go to look at taking a representative sample of this lot, we want to make sure that we retain that same 20% of white colored items in that sample. So the next part of that slide then is a smaller blue box that has 5 items in it. One of those is white color, and that would represent 20% of that having a white item in there. So that concept, if you keep that in the back of your mind as we go forward, that's what we're really fundamentally trying to achieve. And if we put other colored balls or dots in that sample, that's not a good thing. And if we don't have the same percentage of what's in the lot to the sample, that's not a good thing also, and that means that it's unrepresentative. And excuse me for the controls here a little bit. So as we go forward in terms of representative in the form of creating a sampling plan, the government guidance helps us in a few ways, but it's based on statistics. So if we take a look at the statistics, the smaller the number of samples, the lower the statistical confidence is going to be. And you don't have to go all the way back to your statistics class really to worry about this too much. It's basically just a simple fact. If you take one or a small sample, you're not likely to catch everything that's in that statistically. And the more you take, up to and including the whole lot, you could expect a lot higher confidence in the statistical correctness of that sample. So in the industry, in the food industry then, the term N60 is floated about for a variety of reasons, but as you can see, if we take 60 incremental samples from a lot, the probability of detecting something in that lot is about 95 to 96%. If we only took one sample of that, our probability of detection is 5% or possibly even less. So the more samples, the better is the bottom line. And in the standards or guidance of many of these federal guidance anyway, the N60 is really what we're looking at to get a good detectable and representative sample. So if also a secondary takeaway would be if you don't have access to these already, your food safety and quality assurance people in your facilities will likely have this. If not, here's a ready reference for you. The Bacteriological and Analytical Manual is a very good resource. And then the Investigations Operations Manual is what the Food Safety Inspection Service uses it as for their field inspection team. That's a real good one because that's what they're going to be looking at your processes with to determine if you're in compliance or not. So I found that very useful. I think you do too. If you're in that section of the business, I'm sure that's one of your go-to resources. So in those guidance documents then, there are basically three categories that are referenced, at least from the FDA perspective. And that would be that in food category 1, these would be items that you're not going to further process to kill the bugs, if I may use the term a bit loosely. And therefore, you want to do a good number of samples on that, and they recommend an analytical unit number of 60. That way, you have a good idea whether you've got contamination in that product or not. Food category 2, this would be foods that would not normally be subjected to a process to the bacterium, but between time of sampling and consumption. So 30 units are about half the number of the same. And then the category 1 are foods that would normally be subject to a process lethal to the bacteria, and you can expect a decent result with 15 samples in that. In the corner of that last category, I think it's important to note that you should validate this with local FSQA and Food Safety Inspection Service. So internal to your organization, make sure that you are doing methodology that corresponds to what you can expect when the Food Safety Inspection Service team member comes out to take a look at your facility. And that's real important to make sure that you've selected that right and given them a good confidence that you're doing the right things. So in terms of the total mass of samples, the guidance is important, but when the food inspector comes, he's looking for you to prove to them that you've done due diligence. And it's really application-based. So there's not a real fixed, there is guidance, but there's not really a fixed number. If you take some more samples to give your data confidence, that's quite acceptable as well. And it's also pathogen and application specific, as we saw about the different food types. It depends on what types of foods you're processing, what the inputs are, and so forth. And as I mentioned, I don't think I can state it enough that it's very important to look, your food safety and quality assurance people need to be intimately involved in that process of making sure that you're finding what you need to find and assessing those risks properly. Because when FSIS people come, it's going to be important for you to be able to prove to them what you're doing. So let's move on to some methodologies now, and this is kind of the nitty-gritty of how we physically take a sample. And I noted from the feedback on the questions that we asked that a good percentage of you are taking manual samples. And of course, there's a lot of guidance, depending upon what the product is, that there are perfectly acceptable methods, perfectly acceptable ways to do a manual sampling method. But there's some issues with that, and we'll go through those as we go forward. Some are also using completely packaged products, and there's some downsides to doing that as well. And then it looks like there is a significant portion of the listeners today that are also using auto sampling. That's what we'll focus on, is some of the big upsides and advantages to automating the sampling process. So some of the downsides to what we're calling direct sampling, and that would be things like using a scoop or a dipper, or perhaps just a manual valve on a tank, is that you have sample size variation. So if you have two operators that go to take a sample with any of these devices, you're going to have a difference between those two, because it's very difficult to control human behavior, of course, and then your sample size can be in question if one operator takes slightly more or slightly less, and so on. There's just some inherent bias by doing things this way, and one's going to be location variation. So in the case of a valve on a tank, it's maybe not too big of an issue, but if you use a dipper or a scoop, that kind of methodology, the place in the process stream that you actually take that sample then can greatly impact how you do it. Again, using the example of two operators that are going to take a sample. One may do it in exactly the prescribed location that, say, a work instruction shows to take it. Another person would be, say, 20 degrees off of that. The sample is going to just have bias in it from that one operator to the next. And I think that covers the point about human and equipment interactions as well. So you're physically opening up your process to the environment and the operator when you're doing this. So that also leads to the point about contamination. So as you're doing that, there's a lot of opportunity for things to possibly get in and bias the sample. So if there is perhaps old product on the valve or on the scoop, that can certainly go in to contaminate your sample and perhaps give a false positive or even a false negative. In the final comment, you see the Skippy peanut butter product example there. That is one of the methods you mentioned you use, which is final product check. And that's quite acceptable by guidance, and certainly logical in terms of if it's statistically representative of the process you do. But there's some downsides. Obviously, you've got to either hold or retain part of that lot. You need to also dispose of that properly. That's product that's not saleable after that. So there's some downsides to doing that method. One other point that I want to make about that method too is that you don't have the ability to check incremental places or points within your process stream when you do that method. So if you have, say, more than one input into the process, or perhaps more than one operation, you can't really go back and say, well, this part of my process is causing a potential risk or hazard to the product, or if this input versus another input have an impact on the product. So all you're doing is saying that that entire line then has some problem with it, and you still have to go back and try to identify where it is. So the indirect method, and I put alongside of it automated, because in most cases, indirect methods just lend themselves very well to automation. They eliminate some of this imprecision because you don't have the human element in there that's very variable. So you can very easily start and stop the sampling process when the overall process starts and stops. For example, if it's a packaging operation, you can start and stop that sampling process when the product is moving. It allows for composite samples as well. So instead of taking one gross sample and then subdividing it perhaps in a laboratory, you can actually build up your total sample volume by taking smaller individual samples and building up a lot better statistically correct sample. They can be fixed volume grabs too, and that allows you to very precisely control the amount of material that you collect, and then that can build up that composite. And another aspect of that is that you can make it flow proportional. So if your process, especially in liquids, has variability in the flow of the product, if you try to take that manually through a valve, you're going to have bias or imprecision in the representativeness of the sample, because if the flow rate is different when the operator takes a sample, obviously the amount of fluid and or perhaps even the constituents of that are going to change. So you can do flow proportional sampling when you do automated indirect sampling. So another benefit is control, that we can control sample bias. So you can select a location that makes most sense, and I'll have an example of that coming up here. But the bottom line is that if you do that, it's can be very conducive to, for example, taking a sample point just after an input. That would help you identify any problems with that source of input if that were a product or ingredient that you're mixing with it. And of course, it's repeatable. And finally, the indirect method helps to prevent contamination. So the sample itself can easily be isolated from the environment and operators. So those are potential sources of contamination. And that's a real big benefit if you're worried about low level of contamination getting into a product. So let's talk a little more detail about this in terms of sampling concepts then. And regardless really of if you have an automated sampling point or a sampling system or even manual, the place that you choose to get that sample from is very critical. It's very important. And again, it has to do with you know, what's going on in your process, and then also some other things here I'd like to mention. So it's a process point where the process is, or product, sorry, is homogeneous or well mixed. And in liquids, in say high viscosity fluids and that, you want to make sure it's happening after a mixing function and hopefully where the product is very well mixed. So another example I think I mentioned just a few seconds ago was that a good point would be after an input but before final packaging. Also, a good quality sample point would be one that facilitates access to the sample itself, so the operator can easily get at the product and without a chance of contamination. And then it facilitates access to the sampler for maintenance. And again, where the product is moving is a good choice because that usually means that it's homogeneous. It also means that you can take that representative sample incrementally and get a better composite of it. I did mention in terms of liquids, a vertical leg is preferred because gravity works to our advantage in that case to help keeping the product mixed. In strictly liquid applications, if you do a horizontal leg, you need to make sure that's after either a mixing spool or a mixing operation because different components such as water could settle out and then you may not get a representative sample from that. So here's a good example of someone automatically sampling peanut butter. And the bottom line is you can see that the product is easily recognizable. The fill rate on the sample container is recognizable, and the sampler is accessible to the operation people. So let's talk about some applications now. And these are ones that our company has addressed, and we have quite a customer base that does use our solutions in terms of taking samples and have upgraded from a manual method to a more automated method, which has helped them. So some of the methods are quite interesting in terms of, I'm sorry, and pardon me, I'm going to go back a slide. I missed one slide. Other considerations on the sample point are the hygiene before and after sample collection. In many cases, the products, especially in the dairy sector, have specific guidance on what operators should do before taking the sample and after to make sure that the sample is not contaminated. The sample container, of course, is very important to consider that it has the right amount of volume to capture the sample and that it's been decontaminated and is sterile before you take a sample. The documentation part is important too, and that's the real important part as we talk about complying with the Food Safety Act, that the inspectors are going to come in and make sure that your documentation is bulletproof, so linkable from start to finish, and that's very important. Next, we mentioned here a little bit about transporting the sample to the lab, and that the hygiene of that, temperature control and custody transfer between the operation side of things and the laboratory is consistent. Cleaning the sampler between batches is important too, so that you don't have any cross-contamination. And then finally, this is something we'll mention a few more times in the presentation, is validation. And basically what we want to do is make sure that the sample that we take is actually giving the correct data, and your FSQA people will have ways to do that, and there's also guidance about how to test that in the lab as well as the physical samples. And that's good to do internally, obviously, before a third-party inspector or perhaps the Food Safety Inspection Service comes out. Okay, now we can go into some examples. So one very common one is actually in the federal documentation as a perfectly acceptable sample methodology for taking milk samples. And this is sort of a subset of that guidance, which is you need to fully agitate the milk, and there is some time associated with that. You take the sample with a dipper, and that is a fixed volume, but it's pretty difficult to manually take a fixed volume sample. And that's really the methodology when the trucks or the producers go out to the farms to go collect milk. So on the pro side of the equation for this methodology, it is in the federal documentation. It's simple and inexpensive, but the downside of this is that there's quite a bit of opportunity for operator bias. The homogeneity or how well mixed the product is in question, depending upon if the farm's mixing system is good or it's manually done, if that's done properly. And then there's a lot of opportunity for sample container contamination, so that transfer, custody transfer from taking it at the farm and bringing it back to the lab. And I will apologize in advance here. My mouse is not working too well, so I'm going to use the keyboard. Anyway, so a better method possibly to do this milk sampling when we're collecting milk from co-ops or farms is to automate that process. And using a fixed volume sampler, this could be accomplished. So basically, when the milk is being transferred from the farmer's tank to the transport truck, when you start the pump, the sampler could start taking incremental samples of the product. Just as mentioned before, it's flow-based and can be a very nice integrated sample. There's a lower risk of contamination because you can, the operator can load the sterilized sample container prior to starting the process, and it's very easy to keep that relatively low risk of contamination. There's less operator involvement. No need for the mixing to happen because the pumping process helps to provide that. And of course, it's automated, so there's a lot of upsides to this too. You can integrate this with a time date stamp, labeling of the sample containers, and so on for easy traceability down the line. And of course, they're discrete fixed volume grabs, so statistically, this is very accurate way of doing things. So the downside of this, obviously, is that there is a little higher capital investment, but overall, the sample is a lot better. So at the dairy receiving side of things, there's another methodology that's kind of standard practice these days, and that's aseptic septa. And the pros of that, of course, are low cost and that it's an industry standard. There are some downsides to that, though, and the asterisk there is talking about the fact that while the equipment is low capital cost, it doesn't talk about the consumables and how you manage that part of the value stream. So you need to buy the syringes and, of course, replaceable septums, and how you maintain that and keep it clean and so on is important to consider as you use this. So again, there's operator bias. Again, volume can be called into question if one operator doesn't take quite the same amount as another. It is prone to contamination, so it's very contingent upon the operator doing the right things, putting the gloves on properly, sterilizing the septa before taking the sample, and so on. And as mentioned earlier, it does require consumables to come into the equation. So again, another pretty much the same methodology that we use for taking the farm to truck samples. We have the same kind of methodology available, a fixed volume automated sample process for the receiving side of a dairy as well. And if I can get this to work, I'll try again. I apologize. I think I should get it to work. Yes, there we go. Okay. Well, it worked before, but the bottom line is that this air-actuated piston would enter the process pipeline and take a fixed volume based on the annulus, or this volume that you see here in the sampler. Then that discharges it safely into an isolated sample container, and that gives us all the properties that we mentioned earlier. So it's fixed volume, it's automated, so it's a repeatable, very repeatable fixed volume. And then it also drops it into an isolated container, which helps to control the possibility of contamination. And that's what it looks like in the field. So again, on the receiving side of the milk process, when the truck comes up to deliver a load, we can be automatically starting the sampling process and taking that for the custody transfer, as well as the laboratory analysis of that milk. So again, the pros of that are everything that we mentioned in the on-truck sampling method. You have the flow-based fixed volume integrated sample and lower risk of contamination because there's less operator involvement. The whole process can be automated and even include things like time and date stamp. So on the downside, there is a capital investment to consider. So moving on, there's another application I'd like to mention is high viscosity sampling, and the example is peanut butter. So there's obviously a lot of high risk parts of the peanut butter processing application, and using an automated sampler is one of the ways to improve that process. So the bottom line is it's really doing the same things we mentioned on the liquid milk sampling, but in this case, the sampler is actually able to deal with the product that's up to 250,000 centipoise. Again, it's got all the same advantages, and it is automated, and you can go and build up a very representative sample in the process prior to packaging, which is a good way to identify those hazards that might exist in the middle of the process. And I don't think we'll try and see if this works. Yeah, and I apologize. There's a video that shows how that's working too. And again, in process, this can be located wherever it makes sense to be able to access the sampler and, of course, the sample product. So I wanted to move on just a few more examples in here, and we'll get close to wrapping up. In terms of powders, such as protein powders and milk derivatives and so on, there's another methodology used which is also automatable or automated, and that is an auger style sampler. And that can be used to capture dry materials in consistent volumes, quick and easy to clean as well, and it provides pretty much all the same features that we talked about in any of those previous examples. Finally, one last example here where we've got a pet food sampling application. It's an additive for the pet food industry. And again, it's a hygienic sampler that's easy to keep clean, and it's automated, and also is usable with product that's larger in size. So rather than being a powder, for example, or a liquid, this is able to handle larger chunks or pelletized product as well. So in summary, and I apologize for going quick there, and also the fact that we couldn't get those animations to work, representative sampling. So if we do that right, it really helps us identify and mitigate risks because we have a higher confidence level in what we're doing. The indirect sampling methods reduce sample bias and the contamination risks we're talking about. And then the automation of that sampling process gives us a lot of additional benefit that includes integrating it with the process so we don't need to worry about operator intervention, provides a better quality composite sample, and then can be integrated with data acquisition systems for record keeping. So that helps us in many ways in terms of compliance with Food Safety Act. So if I were to give you kind of the big high-level view of what doing sampling correctly and representatively means for us as we do our work, it ensures food testing accuracy. So you can have a higher level of confidence in that data. Increases lot-to-lot repeatability, increases process efficiency, especially on the sampling side of your food quality and safety people, food safety and quality people, reduces process waste, aids in identifying risks more quickly, and simplifies the food safety compliance aspect of it, and ultimately increases profitability. So as I mentioned in the beginning, if there's one takeaway I hope you come away with, that would be that representative product sampling increases confidence in your laboratory data, helps you assess those risks and mitigate those risks in your process. And with that, I am done with my comments and open it up to questions. I think, Nima?
All right, well, thank you very much for your insightful presentation. Now, I'd like to invite our audience to continue sending in their questions or comments using the questions window for the Q&A portion of this webinar. I've already received a few, so we'll start with those. So our first question from the audience is, what does it take to transition to using a cross-cut sampling technique?
Very good question, Nima. A cross-cut sampling technique is really, especially in solid materials, is the preferred method, because that, again, most statistically representative of the product. So taking a complete cross-cut. And to do that depends what you're doing now. If you're doing it manually, obviously you need to take a look at what the total lot volume is going to be, and then what a proper subsample mass or volume is going to be of that. Once you've determined that, then the size and specification of the equipment comes from that. So essentially, we need to determine the type of product where in the process you want to take the sample, where it's more feasible to do that, and not only where it would be more statistically correct, but where you physically can install it, because this has some physical attributes you need to consider. And then the amount of product that the lab needs to get at the end of the day. So those would be the primary things kind of in series that we would ask and do to identify that. And once that's selected, then obviously you would go back and do a validation that would show the relationship between the sample that you're taking and your lab results.
All right, so our next question here is, how do I know whether sampling my finished product is representative or not?
That's also a very good question. There is some guidance from the government, and also hopefully your FSQA people can help, but there's some references in here that you might want to take a look at too. But the bottom line would be that there are guidance, depending upon what you're looking for, that you can do to find that out. And it would be basically a positive result versus a negative result on the lab testing. And there's laboratory methods they can use to put a non-hazardous product that's traceable in the process and find out that, again, in that very beginning slide where we showed the boxes with the particles in it, that if there's 20% or a positive or a negative amount of some contaminant, that you get that at the end of the day. So if your jar you take to the lab at the end of the day has that same quantity in there, then you know that your sampling process is correct.
All right, thank you for that answer. Our next question is it easy to clean your samplers?
Yes, there are, if you take a look at, if you're speaking specifically to Century products, you can take a look at our website as well as YouTube, and we've got quite a few presentations there that show exactly that, how they're serviced and maintained and cleaned. And depending on the sampler, obviously the peanut butter one is a little more difficult, but that is easy to take on and off of the process, and most of our customers would use that in a batch and then be able to do the cleaning process to it very easily by removing it using a tri-clamp type situation. The liquid samplers are able to be cleaned in place in many cases. So the answer to the question is it varies, but yes.
All right, so our next question is, are your samplers certified for use in food sampling?
Yes, most of our samplers that I showed you today are 3A certified, and of course have all that traceability with that too. And the only exception would be the sampler that was the HRX, which was for the large particles. That is considered hygienic, in other words, easy to clean and not susceptible to as much, but it does not exactly hold a 3A certification. The other products do.
All right, well, thank you very much for those answers. We've reached the end of our question and answer portion for this webinar. If your question wasn't attended during the Q&A session, the team from Sentry Equipment will do their best to follow up with you after the webinar. Now, please do take a look at our references guide for more information on this webinar. Thank you everyone for participating in today's conference. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now, please join us in thanking our speaker, John Pobolish, by following the link in your chat box. We hope you found this conference informative. Have a great day, everyone.
Thank you for attending.